Hi, my name's Kerry, and I'm one of the Guardians of our Rivers conservation officers at Buglife, and I'm here today to talk to you about the Riverfly and Monitoring Initiative survey in Scotland. Buglife is a UK-wide charity set up in 2000. We're the only organisation in Europe that is concerned with the conservation of all invertebrates, from bees to beetles, worms to woodlice, and jumping spiders to jellyfish. Buglife's main aim is to halt the extinction of invertebrates and to achieve sustainable populations of invertebrates in the UK. We're working hard to achieve this through promoting environmental importance of invertebrates and raising awareness of the challenges to their survival, assisting in the development of legislation and policy that will ensure the conservation of invertebrates, developing and disseminating knowledge about how to conserve invertebrates, encouraging and supporting invertebrate conservation initiatives by other organisations in the UK, Europe, and worldwide, and undertaking practical conservation projects that will continue to achieve our aim. So, Guardians of our Rivers. We are the hub for the Riverfly Partnership in Scotland. We deliver training and support to community groups so that they can monitor the health of their river by surveying freshwater invertebrates. We're in the second phase of our project, Guardians of our Rivers Next Steps, which is lottery funded. We've been able to employ a part-time conservation officer to join the project. They joined last year. We train new groups and continue to support our existing groups. And we're upskilling experienced volunteers with the advanced training in the extended survey. And we'll soon be training a network of trainers, volunteer trainers throughout Scotland. So there are three main reasons that people get involved with Guardians of Rivers. Restoration. This picture for the restoration is showing the River Mick, the Spittle of Mick in the Cairngorms National Park. And here we have the Cairngorms National Park volunteer rangers from the Balmoral Estate and the River Dee Trust have worked together to plant trees to create much needed shade, and they've installed large woody structures in the river to create habitat. Photo in the middle, pollution. Pollution and water quality is a very popular reason for people to get involved in Riverfly survey. It's becoming, it's in the news quite a lot, and people are really aware of the health of their river. And then there's community. We have people coming together for the training and to go out and monitor their river each month. So why survey invertebrates? Freshwater invertebrates play a vital role in maintaining clean water. They help to break down organic matter and filter organic matter, and they provide a food source for fish, birds, and mammals. And their presence is an indicator of the health of freshwater habitats. They're sensitive to changes in water quality, habitat diversity, water level, and flow. At Guardians, we train groups to do the Riverfly Monitoring Initiative survey. It is a citizen science project to monitor the health of their rivers and detect any decline. The survey is based on a low-level identification of 8 invertebrate groups and uses a simple scoring methodology. And this simplicity ensures a robust and reliable method that can be taught in workshops and consistently applied by trained volunteers in the field. The 8 groups have been chosen as they're sensitive to pollution and stay in the same location and are broadly present all year round. So a decline in their numbers suggests a problem with the water quality. We deliver the training in 2 parts. We have a theory session that is a presentation that we deliver over Zoom, and then there is a practical training session that is in person at the monitoring site at the river. And both of these sessions are around 3 hours. Once the training is complete, our groups of volunteers monitor their site monthly from April to October. And the sampling method they use is a 3-minute kick sample followed by a 1-minute hand search. And this is the same method and the same equipment used by statutory bodies such as the Scottish Environment Protection Agency and the Environment Agency. At each survey, this kick sample is put into a tray, and then the volunteers look for those 8 target groups. They estimate the abundance of these 8 target groups, and then a score can be assigned. So the higher the abundance of a group, the higher the score. And these abundances are entered onto an open access database, which is hosted on a website called Cartographer. This is accessed by anybody. We know that many people are looking at this data. It's been used by students, it's been used by Scottish Environmental Protection Agency, Scottish Water, and many others. So the 3-minute kick sample, we get on our PPE, we wear our waders, gloves, and our buoyancy aid. We use the standardised kick net, and we take a 3-minute kick sample. We disturb the sediment by kicking, holding the net downstream of us, and the invertebrates and the lighter sediment floats into our net. We do this kick sample in a stretch of river, moving our way upstream, kicking in the various habitats. That's because there are some invertebrates that like to live in silty areas, some that like to cling to vegetation, some that like to cling to stones in fast-flowing areas. So we survey these different habitats proportionately. So a 3-minute kick sample will take more than 3 minutes because the timer is only going while we're actively kicking. While we're walking between those habitats, the timer isn't going. Once we've done our 3-minute kick sample, we do a 1-minute hand search where we pick up some rocks in areas where we were unable to kick, especially if the river has some larger boulders. So you can imagine picking up a stone in your garden, you're going to find some things underneath it. So we'll do the same in the river. Just pick up, move a stone and try and get any of the invertebrates we've missed during the kicking. So this is the guide that our volunteers use to identify the 8 target invertebrate groups. First of all, we've got the caddisflies. They are the order Tychoptera, and they're named for the hairy wings of the adults. And these are split into 2 groups. We've got the cased caddis. You can see here the cases vary. They're made out of material found on the riverbed. They can be stones, shells, sand. They can be plant material. So they're all different shapes and sizes. The material was all bound together using a silk and thread excreted by the caddisfly. We've got the caseless caddis. They don't live in a case, but they will make a case to pupate in, and they can also use the silk and thread to create a sort of net that they can use to filter feed. Next, we have the upwing flies, the Ephemeroptera, and these are named after the short lifespan of the adults. Adults don't have functional mouth parts, so they build up all their energy as larvae. So the health of the river is really important, so the larvae can build as much energy as possible to give the adult stage as much chance as possible. So first of all, we've got the mayfly, the Ephemeridae. Then we have the blue wing olive, the Ephemeralidae, the flat-bodied, the Heptageneidae, and the olives, the Betidae. So you can see here we've got lots of tips for how people can ID them, but we also go over things like how they move when we're in person teaching them. And then our next group are the stoneflies. These are Plecoptera, and this is named for the hard, shiny wings of adults. Adults are poor flies. You might find them on a fence next to the river rather than flying around. And then we have the freshwater shrimp, the Gammarus. Of the 8 target groups, the Gammarus spends its whole life cycle in the water, whereas the others, this is the larval stage and they'll emerge as adults. So the benefits of Guardians of Rivers. So our volunteers are contributing to a UK-wide long-term data set, and as well as observing trends, they're picking up any potential issues of water quality. We've been working with SEPA this year, and we were able to provide our groups with site-specific trigger scores. And this is a minimal acceptable score for their site. You can see from the figure that this score fits roughly on the boundary between moderate and poor ecological status. Ecological status is an assessment which is based on biological, physiochemical, and hydromorphological features of the river. The biological assessment looks at communities of invertebrates and plants and fish. The physiochemical assessment looks at the nitrates and phosphates, for example, and the hydromorphological assessment looks at how natural the river is. If the score drops below this minimal acceptable score, the trigger score, it indicates a problem with the water quality. However, this spring and summer, we found that these scores, low scores, were exacerbated by the low flows and the high temperatures of the water. So when this happens, when the groups find that their score has breached their trigger score, we pass all information over to SEPA. This gives citizen science, this empowers our citizen scientists, means that their data is really valuable. And they're getting to work in and around the river, getting to know this hidden world of invertebrates, learning new skills and providing valuable data. And there's groups that have reached their trigger score have been happy with the response so far. It's all very new, but so far we found that the knowledge sharing and conservation conversations with their local SEPA ecologists have been greatly appreciated. Here I have some case studies. So this first one is a catchment scale example of monitoring happening on the Midlothian Esk. So the Esks, the North and South Esk, start in the Pentlands to the south. They join around Dalkeith and then they come out into the Forth at Musselburgh. This site, the Riverfly on the Esk group, was set up as part of a community engagement project back in 2019. And 6 years on, they have 17 sites and over 50 volunteers trained. And as well as doing riverfly survey, they use a basic water monitoring kit to test for nitrates, phosphates, and turbidity. They've also worked with a landowner to plant 230 trees along a problem stretch of bank. From the graph, you can see the green line is the score over time, and that red line is that minimal acceptable score, the trigger score. And there have been a few near trigger events where the score dived down to sit on the trigger level. The local coordinator could tell that all was not well with their river, and they contacted SEPA and found that there was a fault at the local water treatment works just upstream of the site. And following month, the results showed that the score had returned to normal. Another event, a pollution event, was reported to SEPA, and the volunteers downstream of the pollution incident went out to take samples so they could track how far the pollution had moved. And luckily, it was only detected locally. So this just shows how catchment scale monitoring can pinpoint and record the journey of pollution. My next example is on the River Eden in Fife. The trigger score for this site is an 8. And you can see that in September, the score dropped to 4. The site had triggered before, but this was the lowest score that the site had had. The group were concerned because they had noticed the engineering works on the riverbank upstream, and they were worried that it had impacted their site. This information and photos were sent to SEPA, and ecologists went to the site and they were also concerned. So they passed everything on to the SEPA compliance officers, who conducted a site visit of a site inspection of the engineering works to ensure that they had all their relevant licenses in place for the conditions for the works. They also advised the landowners of any additional mitigations that might be required. So the Riverfly Monitoring Initiative is a useful tool for detecting drastic declines in water quality, but for groups that would like more nuanced data, they have the opportunity to upskill. So for experienced volunteers, they can gain more information from their surveys by doing the extended survey training. This provides information on the impact of silt and flow as well as water quality. So at this survey, we look at 33 invertebrate groups rather than 8, which sounds like a really big jump, but quite a few of those invertebrate groups they'll be used to seeing because they've been surveying for a while. Things like beetles, snails, worms. There are a few groups in there that require a bit more skill to learn how to ID. So to do the extended survey, our volunteers will do a self-guided theory session, which is around 1 hour. And then we will come to their survey site and do a practical session. So this will follow a very similar format to the Riverfly Monitoring Initiative training. However, we divide, so rather than doing that 1 minute hand search at the end, we divide that time into a 30-second sweep of the surface of the water, followed by a 30-second search of the rocks. And this is because this survey includes invertebrates that are surface dwelling. So once you've taken out the sample, we'll go through and we'll look for our target groups. And we're unlikely to find all 33 taxa in the survey, so we bring along a reference collection. At the end of the survey, the groups do an ID quiz based on that reference collection. In the photo here, there's a group working through the reference collection together using the guides and hand lenses to identify those 33 groups. The extended survey can take a little bit longer, depending on the site. If the site has a lot of invertebrates, it can take longer. So we do recommend that groups do the extended survey seasonally rather than monthly. But it doesn't replace the Riverfly survey because the 8 groups can be extrapolated from the data of the extended survey. So here are just a few of the taxa recorded in the extended survey. As you can see, it includes beetles, worms, leeches, flatworms, as well as those upwing flies and caddis flies. So what are the benefits of the extended survey? So you're likely to find the survey includes most of the invertebrate groups that you're likely to find in a sample. So that's really good for people that want to know what all the invertebrates that are in their water, in their river. People really enjoy being able to identify all the things that they find. It includes invertebrate groups that are tolerant of organic pollution and low flows and siltation. So it gives us a more nuanced idea of what's going on. And you can see from the figure that the scoring system can be negatively weighted. So here we have non-biting midges. At this site, they were found 200 of them. Non-biting midges can tolerate silt, they can tolerate low oxygen, and they can tolerate pollution. So therefore, their presence in large numbers doesn't necessarily indicate a good water quality. So you can see there that the water quality and the silt and flow score are negative numbers. And these surveys are really useful for monitoring the impact of river restorations and outfalls. They're also being used by groups that are interested in monitoring the impact of agriculture, forestry, and construction. So you've had a lot of great feedback from our volunteers. Here's just a few of them. My favourite is the one in the middle. This is the second best day of my life. This was a group of anglers up at the Aberdeen on the River Don, and I never actually got around to asking what the first best day of his life was, but I was very honoured that this was the second best day of his life. So how could you take part? If you are in Scotland, you can email me and we'll find your nearest Guardians of Our Rivers group. And if there isn't one, we can help you set one up. Our training starts again in spring, and once you're trained, you can go out and do monthly surveys and receive ongoing support. In the rest of the UK, very similar, but rather than emailing me, if you could email the Riverfly Partnership, they will connect you with your local hub so that you can join them for training and surveying. Guardians of Our Rivers groups come in all shapes and sizes. We have individuals who are interested in getting outdoors and reconnecting with nature. We have people that have come together through climate action groups, farming clusters, anglers groups, friends of groups, student groups. Then we have fisheries trusts and rivers trusts and catchment partnerships. Also ranges from local authorities, estates, National Trust for Scotland, Scottish Wildlife Trust and RSPB. And since the project started back in 2022, we now have 58 Guardians of River groups, and we have trained over 800 people. Of the nearly 40 people have trained in the extended survey, and since October 2022, there have been 916 surveys entered into Cartographer. But it's more than just dots on the map. All these dots represent many people spending their time to complete the training and to go out and do the survey each month. And we have been completely blown away by their enthusiasm and commitment. So I'd like to thank all of our volunteers for all the hard work they put in and for their enthusiasm. I'd also like to thank our funders, the National Lotteridge Heritage Fund, and our partners at Riverfly Partnership. Thank you very much for listening.